In this lesson, we will be learning the structure and function of cell organelles in a way that will make it easier for you to remember the varied functions a cell performs. Though cells differ substantially from one another in structure and function, they share common features, such as the plasma membrane, the cytoplasm, ribosomes, and the genetic material. But let's just look at this eukaryotic cell and the labels that it's got. It might seem like a nightmare while learning up all of these structures and their functions. So let's break this down as organelles by function. So I've tried to put this up as a flowchart where we see organelles grouped together for a particular function. So we have the first group where we have the nucleus, the ribosomes, the rough ER, Golgi apparatus, and the smooth ER. All of these related in the synthesis and transport of proteins. In this video, we will focus on synthesis and transport of proteins because that's the biggest chunk of organelles involved and a huge amount of work that's done by these organelles. As we move through the video, I will use analogies for you so that it helps you remember the structure and functions of organelles. So we will begin with the plasma membrane. The plasma membrane is the structure that separates the cell from the environment. All right. And it is a selective barrier because it acts as a boundary wall separating what's inside the cell from what's outside. It's like the boundary wall of an industry, which controls the movement of traffic into or out of that industry. Likewise, the plasma membrane controls what goes in and out of cells, and therefore it is said to be partially permeable. If we zoom into the plasma membrane, this is what you would expect to see, a bilayer of phospholipids. So that's one layer, and that's the other layer. Proteins are found embedded in the bilayer. So that's a protein, and this is also a protein molecule, just like icebergs floating in a sea of phospholipids. Now, why such an arrangement for the plasma membrane? The phospholipid bilayer is partially permeable, allowing only small nonpolar molecules to pass through. They can easily get across the membrane, but large polar molecules cannot pass through the bilayer and therefore make use of channel proteins, these proteins, to pass through the membrane. And proteins in the membrane have other functions too, but that's not going to be covered in this video. Cytoplasm is all the material contained inside the plasma membrane, but outside of the nucleus. It consists of the cytosol and organelles. So what is the cytosol? Cytosol is simply that viscous, semi-transparent fluid. It includes substances like salts, nutrients, some organic substances, all of this dissolved in water. And organelles are the structures that float in this fluid. So you can visualize this like cytosol being the custard and organelles are the fruits in that custard. So most cell activities occur in the cytoplasm. That's the site of metabolic reactions. Now let's begin with synthesis and transport of proteins because that's what our video is all about. Synthesis and transport of proteins begins with the nucleus. Why in the nucleus? We're going to discover that. Before that, let's look at the structure of the nucleus. The nucleus consists of a nuclear envelope, as you see here, and this nuclear envelope has pores. These pores are called nuclear pores, and nuclear pores are responsible for the passage of substances into or out of the nucleus. The nucleus contains the dark, dense structure called the nucleolus that you see here. And the nucleolus is responsible for the synthesis of ribosomes. The nucleus also contains thread-like structures called the chromatin. It is nothing but a complex of DNA and protein, like you see in this image. So this is the DNA that is wound around a group of proteins called histones. And together, it's called chromatin. So that's what you would see ideally in a cell if it is not dividing. But if a cell is dividing, then this chromatin becomes highly condensed, gets packaged into compacted structures called chromosomes. An analogy I have here for you is this. This is what chromatin looks like, and this is what a chromosome would ideally look like. Compacted structure, while a chromatin is tangled up DNA. Okay, so now what are ribosomes? Ribosomes are very tiny structures that are made of ribosomal RNA and protein. So they're made of rRNA plus protein. And they're very tiny structures you could find existing freely in the cytoplasm or attached on the surface of rough endoplasmic reticulum. This ER, endoplasmic reticulum, is rough because it has ribosomes studded on its surface, giving it a rough appearance under an electron microscope. Ribosomes are responsible for the synthesis of proteins. Ribosomes are like the machinery that are responsible for building proteins. So when amino acids join up in a particular sequence, you get a protein. Here's the question. Where do ribosomes get the instructions to make proteins? If I have to give you an analogy here, I would say, let's look at the nucleus as a control room, okay, or a vault. DNA does not leave the nucleus. Ribosomes are out there in the cytoplasm. So there has to be a means of communication. So let's see how this happens. DNA has the instructions. Ribosomes require those instructions for making the protein. Now, DNA, the information on the DNA becomes a protein through two main steps: transcription and translation. All right. What is transcription? Transcription is simply copying the information on the DNA onto a molecule called the mRNA, which is the messenger RNA inside the nucleus. So this process is happening inside the nucleus. Let me just write it down here. It happens in the nucleus. Now, once the information is copied, this transcribed mRNA then moves out of the nucleus through the nuclear pore and then goes on to the ribosomes. So once the mRNA binds with the ribosome, as you see here, it attaches to the ribosome. The ribosome then reads that information and links amino acids in a particular sequence to build a chain of amino acids, which is basically the protein. This process is called translation. So you see how the nucleus, the nucleolus, the chromosomes, ribosomes, they are working together to synthesize protein. So it's, although they seem to be doing things independently, it all piles up to one bigger function. So what next? Once the synthesis is complete, the proteins are ready for transport. So ribosomes, we know, synthesize proteins. The free ribosomes synthesize proteins that are destined to remain and used inside cells. But the ones that are bound on the rough ER, they have slightly different pathway to follow. So first, let's look at what the ER is. We have two types of endoplasmic reticulum. We have the rough endoplasmic reticulum and we have the smooth endoplasmic reticulum. I've already told you why it's called rough, because of the studded ribosomes it has on its surface. The ribosomes on the surface give it a bumpy appearance under an electron microscope, and therefore it's called rough ER. Smooth endoplasmic reticulum does not have ribosomes on it. The rough ER, we know, is a series of interconnected membrane tubules, and the space that they have right here is called the lumen. So the hollow portion of these tubules, this region here, is called the lumen, right? Once the ribosomes have made proteins, the proteins are fed into the lumen. And in here, inside the lumen of the endoplasmic reticulum, the proteins can get folded, they undergo modifications, such as addition of carbohydrate side chains, etc. So the job of the rough ER is to modify protein. Now, remember when amino acids were used to build the protein, so we simply had a chain of amino acids, right? And a chain of amino acids, which is called a polypeptide chain. This protein is still not functional unless it gets folded into a particular three-dimensional shape. So this protein folding happens in the rough ER. That's one thing that happens. Next, glycosylation also happens here, which is addition of carbohydrate or sugar tags to that protein to mark and help process them before transport. And quality check also takes place, wherein any misfolded proteins are caught and they're sent away to be destroyed so that they don't cause any damage to the cell. So this is what the rough ER does. The proteins now need to be sorted, packaged, tagged, and final modifications that need to happen so that they wind up in the right place. So this sorting, tagging, and packaging of proteins takes place in the Golgi apparatus. The Golgi apparatus has two phases. A cis phase, which is the receiving phase, it is near the endoplasmic reticulum, and a trans phase or the releasing phase on the side, which is away from the rough ER, which is closer to the cell membrane. So this end right here is closer to the plasma membrane. The transport vesicles that come from the ER, the rough ER, travel to the Golgi apparatus, the cis phase of the Golgi apparatus. And this vesicle fuses with the membrane of the Golgi. Like you see, one vesicle has fused right here. And then it empties its contents into the lumen of the Golgi apparatus. As the proteins travels through the Golgi, they undergo further modifications. The most common kind of modification that happens here is the addition of short chains of sugar molecules. And once that is complete, the newly modified proteins are then tagged so that it enables them to be routed to their proper destinations. The Golgi is therefore the cargo company or the shipping company of the cell. And that's a very good analogy we could use here. So finally, the modified and tagged proteins are packaged into vesicles. So let's look at what happens to the proteins in the vesicles. Some proteins remain inside and some are released outside. So those that are released outside of the cell are released by a process called exocytosis, like you see here. So the vesicle simply fuses with the plasma membrane. An example I can give you here is the salivary gland cells making the enzyme amylase. Remember, enzymes are proteins. So this protein, or the enzyme here, amylase, is produced by the salivary gland cells, but they're required outside of the cell in the cavity of your mouth so that it can digest food. So this is an example of how a protein made by a cell is released for functions outside the cell. The second thing that could happen to these proteins is they could become incorporated into the plasma membrane. If you can recall, in the beginning of this video, we studied the structure of the plasma membrane. It has a bilayer of phospholipids with proteins embedded. And some of these proteins actually become part of that plasma membrane. Some proteins can also, become those hydrolytic enzymes in lysosomes. So proteins can be delivered to the lysosomes and they can function as hydrolytic enzymes. So hydrolysis is a process in which a large molecule is broken down to simpler molecules. And another function that these proteins can take up is that they can be transported to the smooth endoplasmic reticulum. Now, what's the job of the smooth endoplasmic reticulum and why are proteins transported to SER, the smooth endoplasmic reticulum? Smooth endoplasmic reticulum is this structure that you see here, and it is continuous with the rough ER, but it has no ribosomes on its surface. Smooth ER contains many important metabolic enzymes. And remember, enzymes are protein in nature. and your cell has just made proteins, right? So enzymes are required for synthesizing lipids. Enzymes are required for the synthesis of carbohydrates. And these enzymes have come through that pathway that we've just looked at. Um, Smooth ER also has other functions, which is they help in detoxifying the cell. Let's look at, say, a liver cell. Liver cell would have a lot of these smooth ER because liver cells are responsible for detoxifying medications, poisons, etc. Your muscle cells require calcium ions to trigger coordinated contractions. So your muscle cells would have smooth ER for this purpose, storing calcium ions, which would help in muscle contraction. So that was about smooth ER and a quick summary of the organelles involved in the synthesis and transport of proteins. So we began with the nucleus, right? The nucleus consists of the nucleolus and it has the chromosomes. Chromosomes have coded instructions for making proteins on their genes. Now, this information is transferred through the mRNA to the ribosomes. And who makes the ribosomes? Nucleolus is responsible for making ribosomes. And ribosomes use the information it receives from the chromosomes to build proteins. Proteins are then transported to the rough ER for modification. Once they get modified, they are then transported to the Golgi. The Golgi does the final modifications, and these proteins are finally packaged in vesicles and transported either inside the cell or outside the cell. So this summarizes one of the major functions that our cell does. And look out for part 2, where I will sum up the remaining organelles and their functions.